this video will cover subtopic 3.4, genetic diseases due to recessive alleles. Here are a list of the understandings that we'll cover in this video, as well as the applications that go with these understandings. So with a genetic disease now, this is an illness caused by a gene. Most genetic diseases are recessive, meaning you need to be homozygous recessive to be affected. So you need 2 copies of that recessive allele in order to have that genetic disease. Now, individuals that are heterozygous are considered carriers, so they have 1 copy of that affected allele and 1 normal allele, so they do not display disease, but they do have the capacity of passing the disease to their children. Recessive genetic diseases are typically a surprise, as unaffected parents who are carriers can have affected children. So you can have 2 individuals who are both carriers. So here they have that affected allele, and then they have the normal allele. And the probability of a child having both copies of that infected allele is 25%. A child being completely unaffected is 25%, and it's a 50% chance that any children that they have will be carriers just like them. So the classic example here is cystic fibrosis. So with cystic fibrosis now, this is caused by DNA deletion of a particular gene called the CFTR gene, which is located on chromosome 7. Now, to diagnose cystic fibrosis, it's typically done with a sweat test. So cystic fibrosis is typically associated with high salt levels, so the sweat is tested for its salt content. And genetic tests are done as well to identify this abnormal, to determine if this abnormal gene is present. In terms of treating cystic fibrosis, there's not much that can be done other than altering the diet, the use of antibiotics to prevent and treat lung infections. Lung infections are very common and chronic in people with cystic fibrosis. Other treatments include things like lung transplants and draining of fluids within the lungs pretty regularly, as it builds up with mucus quite a bit. So in cystic fibrosis, you have to keep in mind that normally, in normal circumstances, the mucus is watery. It helps keeps the linings of certain organs moist and prevents them from drying out or getting infected. So it's like a natural kind of cleansing of the lungs. But in cystic fibrosis, the abnormal gene will cause the mucus to become thick and sticky. This mucus will build up in the lungs and will block the airways. This makes it easy for bacteria to grow. It will trap the bacteria, it doesn't clear, the bacteria can grow and can lead to repeated serious lung infections. Over time, these infections can cause serious damage to the lungs. So these diagrams here just show how a normal lung cell would be functioning, and then how a person with cystic fibrosis would be going on in their lungs. So genetic diseases can also be due to dominant alleles, not just recessive alleles. Now, genetic diseases due to dominant alleles represent a small portion of genetic diseases. The majority of genetic diseases are going to be like recessive, due to recessive alleles. Individuals, because this is a dominant allele causing the disease, affected individuals would be heterozygous. And so it is not possible to be a carrier for a genetic disease caused by a dominant allele, as if you have the allele, you will have the disease. So if one parent has the allele for this particular disease, then the chance of the child inheriting the allele is 50%. The example of a genetic disease due to a dominant allele is that of Huntington's disease. In Huntington's disease, the cause is by having too many CAG repeats in this HTT gene on chromosome 4. So this little sequence of DNA repeats itself too many times within this gene and interferes with its normal function. Now, symptoms don't show up until later in life, somewhere between 30 to 50 years old. Odds are by that point, this individual who is carrying this gene may have had a child and potentially pass that gene on to the child. Now, the diagnosis for Huntington's disease. There are several. You have to answer a series of questions. There is a physical exam. There is a very thorough review of your family medical history. If you've had family members, and especially like a mother or a father that has Huntington's disease, that is puts you at a very high risk of having it yourself. And there are neurological and psychiatric examinations as well as genetic testing. The treatment for Huntington's disease, basically there is none. It is incurable and ultimately a terminal disease. So in Huntington's disease, the affected gene, the HTT gene, the function of this particular gene is unknown. However, over the course of the disease, nerve cells do become damaged, causing various parts of the brain to deteriorate. So the disease will affect movement, behavior, and cognition. The affected individual's abilities to walk, think, reason, and talk are gradually eroded. And so the diagrams here shows a typical person's brain versus the section of a brain from an individual with Huntington's disease. You can see the change in the morphology and then the look of the brain itself. There are genetic diseases that are due to codominant alleles that we touched on in the previous video. And so that example is sickle cell anemia. So as we discussed in the previous video, the normal allele for hemoglobin is HB superscript A, and the sickle cell allele is HB superscript S. So an individual who is heterozygous, having both the normal allele and the affected allele, do not have the same characteristics as those who have two copies of the affected allele. So here we have two heterozygous individuals who have children. Here you have a person who has sickle cell disease. Here is a person with who did not get the sickle cell allele, and then these children here, are heterozygous. So they have one normal gene, one abnormal copy of that sickle cell gene. And so these individuals, as discussed in the previous video, do have a slight advantage as they have higher resistance to malaria infection, but do not develop sickle cell disease. Whereas an individual with two normal hemoglobin genes is very prone to malaria infection, or not resistant to malaria infection, but doesn't have any sickle cell traits to them. Now, genetic diseases can also be sex-linked. So most genetic diseases will affect males and females in the same way, but some show a different pattern of inheritance in males and females. So in the previous genetic diseases that we discussed, males and females are equally affected. In sex-linked genetic diseases, the pattern of inheritance shows up differently in males and females, and this is due to the fact that these affected genes are carried on the sex chromosomes, so these are referred to as sex-linked genetic diseases. So the examples for these types of diseases include red-green color blindness. You have males being way more affected than females, and hemophilia. So remember that females have two X chromosomes, that's what defines them biologically as female, while males have one X and one Y chromosome, which is what defines them biologically as males. So when we talk about sex-linked genes, these are genes that are located on the X and Y chromosomes. With sex-linked traits, these are traits that are controlled by genes on the X or Y chromosome. They are identified by their different rates of appearance by males and females. And so this picture here just shows, a Y chromosome versus a X chromosome. So the X chromosome is significantly larger compared to that little Y chromosome. So with the X and Y chromosomes, there is very little homologous DNA between those two chromosomes, which means that the X and Y chromosomes only have a few genes in common between them. The X chromosome has about 2,000 genes, while the Y chromosome has less than 100 genes. For all genes on the X chromosome, females have two copies, as they get two X chromosomes, while males only have one, as they only have one X chromosome. With sex-linked genes now, these can be divided into X-linked genes, so genes specifically found on the X chromosome. And so there's a number of scenarios here that can play out that will determine how children get affected. So if mom is heterozygous for a particular trait, so one allele is found on one X chromosome, a different allele is found on another chromosome, and the dad is not affected, 50% of the children will be affected, regardless of male or female. If mom is heterozygous for the trait and dad is affected, so dad has an affected X chromosome, 50% of their sons will be affected, as you know, they could get a defective or an abnormal X-linked gene from mom, and 100% of their daughters will be carriers, as dad contributes that affected X chromosome. Now, if mom is homozygous for the trait and dad is not affected, 100% of their daughters will be carriers, and 100% of their sons will be affected. If mom is not affected and not a carrier, and dad is affected though, 100% of their daughters will be carriers, and 0% of their sons will be affected, as dad is the one that contributes the Y chromosome to make it a male. So if you want, you can play out all the different combinations of X and Y that would result in these various ratios. With X-linked dominant genes, sons and daughters would need to inherit only one affected allele in order to show a disease. For an X-linked recessive genes, males need to inherit one allele to be affected, that would come from mom, but females need to inherit both alleles to be affected. So these X-linked recessive diseases, or sex-linked diseases, are more common in males than they are in females. And of course, there can be genetic diseases due to Y-linked genes. And so these Y-linked genes are genes found on the Y chromosome. And basically, if a father has an affected Y-linked gene, then all the sons of that affected father will have that trait as well. So looking at some sex-linked traits in humans, so there are a few conditions as we discussed. So there's that red-green color blindness and hemophilia. Now, red-green color blindness and hemophilia are both due to X-linked recessive genes. And the result is that with red-green color blindness, you basically can't distinguish between certain shades. Oh, that's shapes. Shades of red and green. So if you look down at these circles here, you should be able to identify a number in each of these different circles. So there's circles of colors, and in here is a number. So if you have normal color vision, you should be able to distinguish these numbers from the background. Somebody who is red-green colorblind cannot distinguish these numbers, especially the middle one. So, you should be able to see a 12, 57, and what looks like a 74. With hemophilia, these individuals cannot produce blood clotting factors and are very susceptible to bleeding, so they don't clot properly. So that is the end of this video for 3.4. genetic diseases due to recessive alleles.